now we're picking up on the chromosomal theory of inheritance. And so this is bringing in our understanding of modern day genetics, that we could see the cells, we could see that there are actual chromosomes moving around. And we know that those chromosomes are made up of DNA that are carrying the genes that are being inherited. You don't have to know anything about these other people, but just know that there were people who along the way were able to look through microscopes and actually see the chromosomes going through, being segregated and stuff. And so they had the idea that those were the genetic information. But you saw even when we got to the 1950s that sometimes there's still people arguing about what was it, the proteins being carried or the DNA being that was carrying. They were still, took many decades to figure this out. But we know now that inheritance patterns of traits can be explained by transmission patterns of chromosomes during meiosis and fertilization. So chromosomes contain the genetic information. They're replicated and passed from parent to parent. The nuclei of most eukaryotic cells contain the chromosomes in homologous pairs if they're diploid, but their gametes are haploid. And then the formation of haploid cells, the chromosomes segregate independently. We also, that again, they can only do that independent segregation after they're done crossing over and mixing their genes around. And that each parent contributes one set of chromosomes. So in this situation, they're showing that one homolog carries a dominant allele Y for yellow genes, and the other one carries the recessive allele for little y for green genes. And the gametes of the heterozygote may contain the dominant allele or the recessive allele, but not both. They can only get one of them. Again, this goes back to the law of segregation, that they have to get split up. You're not going to make a haploid cell that still has both versions in it. So remember that you have meiosis 1 is prophase. That was where the chromosomes condense down. Metaphase, they actually line up on the metaphase plate. Anaphase, they get pulled apart. Telophase, their nuclei reconfigure to get their membranes back in place. Dissolves down again. We go through meiosis 2 that repeats everything. Prophase, metaphase to line up right here. And then it gets pulled apart again to give us now 4 haploid cells. This would have to happen for all of the chromosomes, though, not just one, as this picture shows. So, because we had more than one chromosome in there, what you get is, in this case, you could have where both blue chromosomes lined up on the left side and both red on the right, or you could have them flip-flopped. And depending on what was happening, you could end up then with blue, 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 red, 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 or red and blue, blue and red, blue and red. Now, on a pea plant that only has 7 chromosomes, that you could still draw out, but don't forget, humans have 23 pairs of chromosomes. So that gives us many possible variants. And remember that each chromosome is carrying hundreds, if not thousands, of genes on it. So lots of stuff happening. And these pictures are not showing where during prophase 1 where there's crossing over. Sometimes you could have this blue and red lining up on here, and these legs would actually cross over to exchange genetic information. So, but the idea here is that independent assortment is occurring because they randomly line up along the metaphase plate. We don't do breeding like this to study inheritance patterns in humans, so we have to draw pedigrees and look at what happens to find out if the trait was dominant or recessive. We've talked about this a little bit, but just a reminder here. Females are shown as circle because girls are curvy. Males are shown as squares. The diamond shape is for when sex is unknown or not specified. They use a smaller symbol that shows miscarriage. A deceased individual has a line through them, a slash. Unaffected individuals are unshaded. Affected individuals are shaded in a solid color. So presumed heterozygotes or carriers are going to be partially filled in. Now you have to be careful on pedigrees because a lot of the times if we don't know who a carrier is, they might not be shown at all on the pedigree. And it might, when we're studying recessives, we go, well, crap, nobody in this generation has it. And suddenly it shows up. Well, that means somebody had it and they must have been a heterozygote carrier, or both parents were heterozygous carriers. It doesn't happen commonly, but sometimes you see a double line here where that's mating between related individuals. And then again, another rare thing that can happen is you could see this upward arrow V shape, and that shows twins as opposed to showing here where it's a straight line. If these two had been twins, it would have been a V shape. So we are going to work with pedigrees more on Wednesday during our bloodlines case study. But just to give you an example here, you could have a disease that has a normal allele and then a mutant allele that causes the symptoms of the disease. So whenever we see a recessive pattern of inheritance, that's when two unaffected individuals are going to have 25% affected offspring. Again, they're showing heterozygous individuals. Well, we can't tell just by looking at something. Are you heterozygous or are you homozygous dominant? And then two affected individuals will have 100% affected offspring. If both of your parents had the disease, chances are you're going to get the disease if it's recessive. Now, dominant pattern of inheritance is when you have the affected individual has at least one affected parent, unless it's a random new mutation. And so the example they give here is cystic fibrosis, which is a recessive disorder in humans that the main problem you see, like the main symptom, is that there's a buildup of sticky mucus in the lungs that makes breathing difficult. This mucus is also prone to getting bacteria in it, and that then leads to infections, pneumonias. It has some other abnormalities in other tissues because it affects, I want to say, chlorine pumps on it, so that the ions aren't moving properly. But you don't need to know the details about it. Just know that it is a recessive disorder that, unless things have changed recently with treatments, had the life expectancies in their 20s. So looking at this pedigree, we see up here on the P generation, two carriers. These are heterozygous parents. Now, if we make our Punnett square, that would look just like the one that I drew before with our big T's. We have 1/4 homozygous dominant. They would be unaffected. 2 out of 4 are going to be heterozygous. So they are going to be carriers, but they don't show any symptoms. And then 1/4 would be a homozygous carrier or homozygous recessive that is going to actually show the disease, actually going to have symptoms. So now, looking at that Punnett square, they had 3 offspring. One of them was completely unaffected, homozygous dominant. They married a homozygous dominant. All offsprings are homozygous dominant, no problems. One of their children is affected because they have homozygous recessive, little t. And then one of the children is a carrier for being heterozygous. They married another person who was a carrier. Now, instead of following the 1-to-1 ratio that we would see, usually they had worse luck and 2 of their children were actually affected. So because these carriers do not show any symptoms, this is why it could be a good idea to have genetic testing done, particularly if you have anybody in your family that has something like cystic fibrosis, there's a chance that you could be carrying the gene and then your new spouse is carrying the gene, but since neither of you are sick, you think everything is fine, and then you end up having children with a disease that gives them a life expectancy of 20. So again, we are going to work with pedigrees more on Wednesday. So on the probability and statistics, they go into way more stuff than you need to know. But the ones that you are going to look at, we've already talked about chi-square analysis and p-values, but we're going to review that one. Just knowing probability is just the number of times it would occur versus the total number of possible outcomes. So in a coin flip, how many times are you going to get heads? You get one heads out of heads and tails, so 1 out of 2, 50%. A lot of the times when we were working on stuff, we'd have a 3 out of 4. Remember, 3 out of 4 is 75%. On our dwarfs, we had 1 dwarf out of 4 possibilities. We get 1/4, 25%. Now, the larger the size of the sample, or the more times the experiments before, the more closely the observed results will match the expected outcomes. So going back with our dog hair, when we're doing chi-square analysis, if you only check a couple dogs, you're probably not going to notice a big pattern. But if you start seeing the pattern when you have more than 1,000 samples, then you know it's something that's going to be a thing to pay attention to. So their example here is that if you flip a coin only 10 times, it's not unusual to get 70% heads, 30% tails. But if you flip it 1,000 times, the percentage of the heads is going to be pretty close to the predicted 50% value. So when they were doing the analysis to find out a 3-factor cross, they did multiplication instead. And what you're looking here is, this is for when your independent outcomes are unrelated to each other. So the probability of 2 or more independent outcomes will occur is equal to the product of their individual probabilities. So in this case, they're talking about a disorder called congenital analgesia that is a recessive trait in humans where they can distinguish between sensations, but they don't perceive extreme sensations as pain. They don't perceive pain, or not perceiving pain is quite dangerous, because if you have somebody who touches something hot, usually your body says, hey, this hurts, pull your hand away. If you have this condition, you're not going to pull your hand away. Or if you could end up breaking bones or having a bad cut and not even know that it's there. So the question here is, 2 heterozygous individuals plan to start a family. Well, this is recessive trait that says these people are heterozygous, then before we can call them carriers. What is the probability that the couple's first 3 children will all have congenital analgesia? So in this case, we could look at a normal Punnett Square of 2 heterozygous individuals, and we get that 1/4 being double recessive. But now, because each child is an independent roll of the dice, we have to go 1/4 times 1/4 times 1/4. 1/64 becomes 0.016, becomes 1.6% probability that the first 3 children will all exhibit the disease. So very low odds. If you only have a 25% chance on each kid, the odds that all 3 got the bad version, very, very slim. In case you wanted to do a 3-factor cross, here's the way to show how to do that without using a 64-square Punnett Square. It's saying, okay, our genotype here, our genotype here, these are the 2 parents. What is the probability that the offspring is going to have this? So we break it down into 1 factor at a time. If we wanted 2 A's and we have heterozygous, we know that our answer is 1/4, because 1, 2, 4. We can say 1/4 for the A's. What are the odds of getting BB? Well, our combination is big B, little b. little b means it's gonna, if you do your Punnett square, you should come out with 50%. If we want big C, little c, and we have big C, big C, big C, little c, when you do the math, you end up with 50%. So now we multiply all of these together. What are the odds that you're going to get this A, this set of B's, this set of C's, and you get 6.25%. Yeah. You do not need to know about binomial expansion equations and how to apply it. You don't need to know about multinomial expansions. Chi-square test, again, we've already talked about this back at the dog hair, and you are not going to be asked to calculate the chi-square, but what you do want to be able to understand is if I tell you this chi-square and I give you the table, that you could look at it and find the p-value. And remembering that the cutoff for if this is statistically significant is when their p-value is less than 0.05, so less than 5% of the time. If it occurs more than 5% of the time, then we go, yeah, that's probably just a random chance. Like, that's not a statistically significant thing. So you can go back and review from there. But in this case, they were talking about the degrees of freedom, and using our chi-squared value here being 1.06, then we can go through and go, all right, that is definitely higher than the 0.05 cutoff. So in this case, it says P of 0.8 means expected to occur 80% of the time based on random chance alone. You want to find it when it is way over on this side. The 0.05, null hypothesis rejected. 0.05 and 0.01, remember in a paper, this would be shown as 1 asterisk. This would be shown as 2 asterisks. So that brings us to the end of the chapter here. Does anybody have any questions, anything you want me to go over? Before I show chapter 4, I'm not actually even going to go through the PowerPoint for chapter 4 right now. I'm going to tell you on chapter 4, the study guide tells you to watch this video, section 4.1 video, sex determination is more complicated than you thought. And that goes through some of the different traits that are different ways that animals determine their genders, with humans being X and Y, birds use Z and W, some creatures use temperature, some use a haploid diploid. You don't need to know the specific examples. You just need to know your specific examples based on like which species use which. Just know the types of possibilities, like some reptiles use temperature, that some insects use haploid diploid. Just know possible choices that can be used. We're going to cover the rest of chapter 4 on Wednesday after we do the bloodlines case study, because that's going to have us practice the pedigrees. But it also works with sex-linked inheritance. I want you to go through, these are lab resources that are virtual labs. So the first one on genetic inheritance, it has you play around with some different traits in a family with pedigree. These are all together, kind of to have fruit fly characteristics. And then once you learn the characteristics on how to sort out the fruit flies and stuff, then you do a monohybrid cross. So again, that's the single factor. Then you do a dihybrid cross. So if you did just wing length here, you might do wing length and body color. And then you have X-linked fruit fly cross, which is probably red and white eyes, I believe. We actually have our flies in our lab that are red and white cross. They are going to be sitting there. Hopefully they'll be at the F2 generation by the time we come in on the 15th. But you are going to play with them in the lab some, so I want you to understand the virtual lab side of it. So you should have a general understanding of how X-linked stuff works because it's going to come up on our bloodline case study. And anything that isn't covered by the case study, I will jump back into chapter 4 to discuss with you. So again, last call for questions. I'm going to stop this recording here.